Hi everyone, this is Mackenzie, one of the theory teachers for General Biology 2. Haven't met all of you, but some of you I have. This is one of the first capsules, if you choose to watch mine first, before Boris's. What's nice about these capsules is that you can take your time and pause anytime that I'm going too quick and you need to write down notes, or you can rewind to see what I said if you miss something. The learning objectives for this capsule are to identify and explain the functions of a cell's plasma membrane, cytoplasm, non-membranous and membranous organelles, as well as recognize the unique structures of mitochondria and chloroplasts. Now, please take a second to pause the video and try to remember what organelles are, as well as recall some organelles that we learned in General Biology 1. In this course, we will go into more depth on specifically the plasma membrane, as well as some of the chemical reactions in specific organelles. But for this capsule, it's more serving the purpose of reviewing of these concepts. Cells are the basic unit of life. This is a really important concept to know. You see that cells are able to respond to stimulus. They're able to adapt to their environments, able to produce energy through chemical reactions, and reproduce. These are all key elements to life. They make up the building blocks of tissues and then eventually organisms when we looked at the levels of organization. It's important to note too that even if an organism were to die, inside of them, some of their cells might still be alive according to those key elements. We also know that they are made from pre-existing cells and that they perform vital functions for our body. Now, in this picture, in this figure, we see a few different types of cells. And this is really just showing the diversity cells have. In our body, we have over 30 trillion cells, but there are only 200 types. And those 200 types all have unique structures and functions. So we see this structure-function relationship at the level of the cell. I'd like for you to pick a cell from the picture, it could be any one, and try to find out its function if you don't know it. Once you find out the function, think about and explore through research, how does its structure enable or benefit its function? So for this capsule, we're going to focus on just a typical cell. Like we had said, there is 200 different types, but we're focusing just on a typical cell to go over the main components. With the diversity that we see, some of these components might be different or missing depending on the type of cell. We have, in a typical cell, a plasma membrane. So this is kind of the outside, which encases the cell. And the cell is divided to the plasma membrane. And then when we go inside of the cell, we get to the cytoplasm. The cytoplasm can be divided into the cytosol and organelles. Then within the organelles, they're subdivided into two different categories as well. Let's begin with the plasma membrane. We're sticking kind of more so just to reviewing the function right now of the membrane. And then next week, we'll venture more into specific details of their functions. The plasma membrane is found in all cells, eukaryote, prokaryote. Its function is to protect and give the cell structure. It also helps with detection. So this might be detecting different stimuli or changes in the environment. It also will control the movement of materials in and outside of this cell. We have already looked at a little bit the plasma membrane and seeing that it is a phospholipid bilayer. But like I said, next week we'll go into more detail with respect to this plasma membrane and its functions. Moving to the inside of the cell, now we find the cytoplasm. The cytoplasm will encompass both the cytosol and organelles. When we look at the cytosol, this is going to contain a mixture of dissolved nutrients, ions, both soluble and insoluble, proteins, as well as waste products. Now, these proteins that we see are most times enzymes that will regulate metabolic operations within the cell. You might also notice from cell to cell, the consistency of cytoplasm differs. At times, it might be as thin as maple syrup or as thick as more of like a gelatin. This diagram is just illustrating the difference between the cytoplasm and cytosol. So the cytoplasm is including everything within the cell, where the cytosol is only including that kind of fluid outside of the organelles. And now we've made it to the organelles of the cell. Organelles are, you can kind of compare them to organs in our body. They have specific functions which are all separate, but they all ensure that the cell is healthy. And some organelles, however, do work together and form almost like an organ system would. They do multiple jobs that together are able to create a common goal. Organelles are internal structures that perform specific tasks. And remember that with each cell, the amount of organelles or the types of organelles found in them may differ. Before we specifically go on to each type of organelle, I'd like you to keep in mind these two cells. So we have over here smooth muscle cells, and over here we have red blood cells. So start to think about what are the functions of these cells. And as we go through the organelles, I want you to think of which organelles would each cell have, in what amounts, and how might they differ. Okay, we are going to start with non-membranous organelles. Just like the name states, non-membranous, that means they don't have a membrane associated with them. Now, depending on the source that you read or watch, they might not consider non-membranous organelles as actual organelles, but we are, because they do have specific functions within the cell. You also may remember that prokaryotes, they don't have organelles. Well, when we say that, we mean that they don't have membranous organelles. They would still have these non-membranous organelles. The first being the cytoskeleton. So this is a nice picture showing all the yellow and blue is showing the cytoskeleton. It's giving it strength and support and structure. Here we have a cell with some villi or microvilli, and those are created because of the cytoskeleton. Next, we have centrioles. These are similar to the cytoskeleton. They also perform or give the cell some support. You've seen them before as those spider web-like spindles in mitosis and meiosis that are responsible for moving the chromosomes during cell division. And they organize themselves into microtubules. So here's just a zoomed in version of a centriole. So they would extend either way, making those long spindly fibers. And lastly, we have ribosomes. These are a protein responsible for protein synthesis. You see these, remember, during protein synthesis, they take or read the mRNA and they turn that mRNA into a polypeptide chain. Just recall that ribosomes have two subunits, a smaller one and a larger one, and they clip on or clamp on to the mRNA when they're reading it. Next, we have membranous organelles. So these do have a membrane. We'll start with kind of the really important one, the nucleus. It's responsible for storage and processing of genetic material. So our DNA is found within the nucleus. It's important because it's kind of like the control center of the cell. It's going to control what proteins are synthesized as well as metabolism. Next, we have the endoplasmic reticulum. This is a continuous system of flattened membrane sacs, and it manufactures carbohydrates, steroids, and lipids. You might remember that there's two types of endoplasmic reticulum. There is the rough endoplasmic reticulum, which we see here with all the little dots. And there's also the smooth, which doesn't have the dots. Now, do you remember what these dots are? These little dots are our ribosomes. So that tells us that the rough endoplasmic reticulum is going to be responsible for creating proteins. Next, we have the Golgi Apparatus. Again, this is stacks of membrane-bound sacs. It also contains many membranous vesicles, and they'll carry proteins and lipids. The Golgi apparatus is most known for the alteration and packaging of secretory products. Lastly, we have lysosomes and peroxisomes. These will remove damaged organelles or any pathogens. So they're kind of the garbage disposals. They have this membranous vesicle, and within them, they're holding on to that degraded garbage. That might include enzymes, organelles, or again, pathogens. Peroxisomes are more known for neutralizing pathogens. So they have enzymes within them that will do that. In the beginning with the organelles, I had said that some work together almost as a system. And an example of this is the endomembrane system. So this system is responsible, a group of different organelles come together and they modify and package and transport different vesicles filled with proteins and lipids. Here's just a short video showing the endomembrane system working together.
The endomembrane system consists of the endoplasmic reticulum and Golgi apparatuses. Vesicles pass from one part of the endomembrane system to another, and secretory vesicles from the Golgi apparatus merge with the plasma membrane, secreting their contents by exocytosis.
So we can just see that popping out or budding out is a membranous vesicle from the endoplasmic reticulum, and then it moves towards the Golgi apparatus. There's two really important membranous organelles or special organelles that we're going to be focusing on in this course. One is mitochondria and the other is chloroplasts. So here we have a mitochondria. They are found in most eukaryotic cells, yes, even plants. Often we think that they're only animal cells because they are where ATP is made, but they're also found in plant cells. They're composed of both an outer and inner membrane, which we can see here, as well as in a real picture, kind of the outside, that outer membrane, as well as an inner membrane. And within those membranes are where some important chemical reactions happen, which we'll go into more detail within the next few weeks. They also have what's called Cristae, and this is just that membrane folding in on itself. So it's this double layered membrane. Within the Cristae, they have what's called a matrix. It's just the inside of the Cristae. And as you might know, mitochondria are known as the cell's powerhouse because that's where we get our energy. They produce ATP for us. Moving on to the chloroplasts. They also have kind of a similar structure to the mitochondria with an outer membrane and an inner membrane. They also have a stroma, that's just a liquid material found within the chloroplast, and cycloids. So these are the structures that are stacked. This is where the site of light-dependent reactions happen. And once you stack a few cycloids together, you get a granum. So that's what we're seeing in this real picture. That stack is the granum. A single one would be a cycloid. Chloroplasts are used in plant cells only to make glucose, which is food for the plant. Now that we went over all the different organelles found in cells, let's think about the two cells mentioned before, muscle cells and blood cells. Now in muscle cells, what's their function? What do they do? Well, they're responsible. There's a few different types of muscle cells, but they're responsible for contracting or moving things. If it's a skeletal muscle, its job is to contract and move our body, like our bones and our arms, legs. If it's smooth muscle, it's used to contract and push food through our body, or also contract for our veins and arteries to dilate, as well as cardiac muscle cell is used to help the heart beat. So we know that they have to contract a lot. And if they have to contract a lot, they're going to have a special cytoskeleton to allow them to contract. So in a smooth muscle cell, we see them having this kind of grid-like cytoskeleton, which when they contract, allows the cell to shrink. In a red blood cell, their function is different. They don't need to contract, but what do they need to do? Well, they're responsible for delivering oxygen. So to deliver oxygen, they need to get through some very small places. Think about the little tiny blood vessels that you see in your eye. They're very tiny. So if they need to do some squeezing, they need to have a cytoskeleton that is flexible enough to do that. So their cytoskeleton are made up of different filaments. So here we have myofilaments with the muscle cell. Here we have something called spectrin. And the way that they create, it's a triangular grid. This allows it to be flexible and move. They can bend through tiny little vessels. So we see differences in the two with respect to their cytoskeletons. What about for organelles? We know that muscle cells, they do a lot of contracting, so a lot of moving. If they need to do a lot of moving, they need to have what type of organelle? They need to have a lot of mitochondria. So within muscle cells, you'll find lots of mitochondria because they need that ATP, they need that energy to do their function. They also have nuclei. So you can see this is a tissue piece from smooth muscle. You can see lots of nuclei within each cell. Now, blood cells, their sole function is to distribute oxygen or deliver oxygen. They don't need to reproduce, so they don't need to, say a muscle cell got damaged, they don't need to reproduce to cover up that damage. They really just need to squeeze through tiny places. So they don't need any kind of extra organelle. So blood cells, if you look at them, you can't see a nucleus because they don't have one. They don't need one because they need to be very flexible. They also don't need to reproduce or duplicate. Another reason why they don't have a nucleus. They also don't have any mitochondria because they don't need the energy. They are pushed through veins with plasma and other components of blood. So here we can see, just by looking at two different types of cells, how drastic their structure is, which also tells us how drastically different their function is. So hopefully by the end of this capsule, it was mostly a review, and now you can both identify and explain the function of the plasma membrane, the cytoplasm, both types of organelles, membranous and non-membranous, as well as recognize unique structures of the mitochondria and chloroplasts. In Boris's capsule, he's going to go into more detail of the mitochondria and chloroplasts with respect to their function. So we covered the structure, he's looking at the function. And I'll see you all, or some of you, on Wednesday. Thanks for watching.